Genetic drift. Genetic drift was introduced when discussing the likelihood of panmixia being widely extant as a natural fertilization pattern. See section on allele and genotype frequencies. Here the sampling of gametes from the potential gametome is discussed in more detail. The sampling involves random fertilization between pairs of random gametes, each of which may contain either an A or an allele. The sampling is therefore binomial sampling. 13, 382 to 395, 14, 49 to 63, 29, 35, 30, 55. Each sampling packet involves two N alleles and produces N zygotes, a progeny, or a line, as a result. During the reproductive period, this sampling is repeated over and over, so that the final result is a mixture of sample progenies. The result is dispersed random fertilization. These events, and the overall end result, are examined here with an illustrative example. The base allele frequencies of the example are those of the potential gametome. The frequency of A is QG equals 0.75, while the frequency of A is QG equals 0.25. White label, 1, in the diagram. 5 example actual gametomes are binomially sampled out of this base S equals the number of samples equals 5, and each sample is designated with an index, K, with K equals 1. S sequentially. These are the sampling packets referred to in the previous paragraph. The number of gametes involved in fertilization varies from sample to sample, and is given as 2 enic at white label, 2, in the diagram. The total sigma number of gametes sampled overall is 52, white label, 3, in the diagram. Because each sample has its own size, weights are needed to obtain averages and other statistics when obtaining the overall results. These are, and are given at white label, 4, in the diagram. Genetic drift example analysis. The sample gametomes, genetic drift. Follow completion of these 5 binomial sampling events, the resultant actual gametomes each contain different allele frequencies, pK and qk. These are given at white label, 5, in the diagram. This outcome is actually the genetic drift itself. Notice that two samples K equals 1 and 5 happen to have the same frequencies as the base potential gametome. Another K equals 3 happens to have the P and Q reversed. Sample K equals 2 happens to be an extreme case, with pK equals 0.9 and qk equals 0.1, while the remaining sample K equals 4 is middle of the range in its allele frequencies. All of these results have arisen only by chance through binomial sampling. Having occurred, however, they set in place all the downstream properties of the progenies. Because sampling involves chance, the probabilities integral K of obtaining each of these samples become of interest. These binomial probabilities depend on the starting frequencies Pg and Qg and the sample size 2 enic. They are tedious to obtain, 13, 382 to 395, 30, 55, but are of considerable interest. See white label, 6, in the diagram. The two samples A equals 1, 5 with the allele frequencies the same as in the potential gametome, had higher chances of occurring than the other samples. Their binomial probabilities did differ, however, because of their different sample sizes 2 enic. The reversal, sample K equals 3, had a very low probability of occurring, confirming perhaps what might be expected. The allele frequency gametome K equals 2 was not rare, however, and the middle of the range, sample K equals 4 was rare. These same probabilities apply also to the progeny of these fertilizations. Here, some summarizing can begin. The overall allele frequencies in the progeny's bulk are supplied by weighted averages of the appropriate frequencies of the individual samples. That is. And. Notice that K is replaced by, for the overall result, a common practice. 9 results for the example are P equals 0.631 and Q equals 0.369, black label, 5, in the diagram. These values are quite different to the starting ones Pg and Qg, white label, 1 inch. The sample allele frequencies also have variance as well as an average. This has it using the sum of squares SS method 31, see to the right a black label, 5, in the diagram. Further discussion on this variance occurs in the section below on extensive genetic drift. Progeny lines, dispersion. Frequencies of the 5 sample progenies are obtained from the usual quadratic expansion of their respective allele frequencies, random fertilization. Results are given at the diagram's white label, 7, for the homozygotes, and at white label, 8, for the heterozygotes. Rearrangement in this manner prepares the way for monitoring inbreeding levels. This can be done either by examining the level of total homozygosis P2, K plus Q2, K equals 1 minus 2 PKQK, or by examining the level of heterozygosis 2 PKQK, as they are complementary. 32 samples K equals 1, 3, 5 all had the same level of heterozygosis, despite one being the mirror image of the others with respect to allele frequencies. The extreme allele frequency case K equals 2 had the most homozygosis, least heterozygosis of any sample. The middle of the range, case K equals 4, had the least homozygosity, most heterozygosity, they were each equal at 0.50, in fact. The overall summary can continue by obtaining the weighted average of the respective genotype frequencies for the progeny bulk. Thus, for A, it is, for A, it is and for A, it is. The example results are given at black label 7 for the homozygotes and at black label 8 for the heterozygote. Note that the heterozygosity mean is 0.3588, which the next section uses to examine inbreeding resulting from this genetic drift. The next focus of interest is the dispersion itself, which refers to the spreading apart of the progeny's population means. These are a C-section on the population mean for each sample progeny in turn, using the example gene effects given at white label 9 in the diagram. Then, each is obtained also at white label 10 in the diagram. Notice that the best line k equals 2 had the highest allele frequency for the more allele A. It also had the highest level of homozygosity. The worst progeny k equals 3 had the highest frequency for the less allele A, which accounted for its poor performance. This line was less homozygous than the best line, and it shared the same level of homozygosity, in fact, as the two second best lines k equals 1, 5. The progeny line with the more and the less alleles present in equal frequency k equals 4 had a mean below the overall average, see next paragraph, and had the lowest level of homozygosity. These results reveal the fact that the alleles most prevalent in the gene pool, also called the germplasm, determine performance, not the level of homozygosity per se. Binomial sampling alone affects this dispersion. The overall summary can now be concluded by obtaining n. The example result for P is 36.94, black label 10 in the diagram. This later is used to quantify inbreeding depression overall, from the gamete sampling. See the next section. Recall that some non-depressed progeny means have been identified already, k equals 1, 2, 5. This is an enigma of inbreeding. While there may be depression, overall, there are usually superior lines among the gamodeme samplings. The equivalent post-dispersion panmictic inbreeding. Included in the overall summary were the average allele frequencies in the mixture of progeny lines, page, and q. These can now be used to construct a hypothetical panmictic equivalent. 13, 382 to 395, 14, 49 to 63, 29, 35. This can be regarded as a reference to assess the changes wrought by the gamete sampling. The example appends such a panmictic to the right of the diagram. The frequency of AA is therefore page 2 equals 0.3979. This is less than that found in the dispersed bulk, 0.4513 at black label, 7 inches. For A, Q2 equals 0.1303, again less than the equivalent in the progeny's bulk, 0.1898. Extended principles. Other fertilization patterns. Spatial fertilization patterns. In these sections, dispersive random fertilization, genetic drift, has been considered comprehensively, and self-fertilization and hybridizing have been examined to varying degrees. To the left depicts the first two of these, along with another, spatially based, pattern, islands. This is a random fertilization featuring dispersed gamodemes, with the addition of overlaps, in which non-dispersive random fertilization occurs. With this pattern, individual gamodeme sizes 2n are observable, and overlaps m are minimal. This is one of Sewell-Wright's array of possibilities. 37. In addition to spatially based patterns of fertilization, there are others based on either phenotypic or relationship criteria. The phenotypic bases include assortative fertilization between similar phenotypes and dissociative fertilization between opposite phenotypes. The relationship patterns include sib crossing, cousin crossing, and back crossing, and are considered in a separate section. Self-fertilization may be considered both from a spatial or relationship point of view. Islands, random fertilization. The breeding population consists of S small dispersed random fertilization gamodemes of sample size k equals 1. S with overlaps of proportion in which non-dispersive random fertilization occurs. The dispersive proportion is thus. The population consists of weighted averages of sample sizes, allele and genotype frequencies and progeny means, as was done for genetic drift in an earlier section. However, each gamete sample size is reduced to allow for the overlaps, thus finding effective for islands, random fertilization. For brevity, the argument is followed further with the subscripts omitted. Previous sections treated dispersion as an assistant to selection, and it became apparent that the two work well together. In quantitative genetics, selection is usually examined in this biometrical fashion, but the changes in the means, as monitored by delta G, reflect the changes in allele and genotype frequencies beneath this surface. On genetic drift brings to mind that it also affects changes in allele and genotype frequencies and associated means, and that this is the companion aspect to the dispersion considered here, the other side of the same coin. However, these two forces of frequency change are seldom in concert, and may often act contrary to each other. 1. Selection is directional, being driven by selection pressure acting on the phenotype. the other genetic drift is driven by chance, at fertilization binomial probabilities of gamete samples. The two same allele frequency, their coincidence, is the probability of obtaining that frequencies sample in the genetic drift. The likelihood of their being in conflict, however, is the sum of probabilities of all the alternative frequency samples. In extreme cases, a single syngamy sampling can undo what selection has achieved, and the probabilities of it happening are available. It is important to keep this in mind. However, genetic drift resulting in sample frequencies similar to those of the selection target does not lead to so drastic an outcome, instead slowing progress towards selection goals. Correlated attributes. On jointly observing two or more attributes, e.g. height and mass, it may be noticed that they vary together as genes or environments alter. This correlation is measured by the covariance, which can be represented by COV or by theta. 43. It will be positive if they vary together in the same direction, or negative if they vary together but in opposite direction. If the two attributes vary independently of each other, the covariance will be 0. The degree of association between the attributes is quantified by the correlation coefficient, symbol R or rho. In general, the correlation coefficient is the ratio of the covariance to the geometric mean, 70, of the two variances of the attributes. 196 to 198 observations usually occur at the phenotype, but in research they may also occur at the effective haplotype, effective gene product, see figure to the right. Covariance and correlation could therefore be phenotypic, or molecular, or any other designation which an analysis model permits. The phenotypic variance is the outermost layer, and corresponds to the usual covariance in biometrics statistics. However, it can be partitioned by any appropriate research model in the same way as was the phenotypic variance. For every partition of the covariance, there is a corresponding partition of the correlation. Some of these partitions are given below. The first subscript G, A, etc. indicates the partition. The second level subscripts X, Y, R, place keepers, for any two attributes. Sources of phenotypic correlation. The first example is the unpartitioned phenotype. The general partitions, A, genotypic, overall genotype B, genic, substitution expectations and C, allelic, homozygote follow A, B, C. With an appropriately designed experiment, a non-genetical environment partition could be obtained also. Underlying causes of correlation. There are several different ways that phenotypic correlation can arise. Sample size, sample statistics, and other factors can influence the ability to distinguish between them with more or less statistical confidence. Each of these have different scientific significance, and are relevant to different fields of work. Direct causation. One phenotype may directly affect another phenotype, by influencing development, metabolism, or behavior. Genetic pathways. A common gene or transcription factor in the biological pathways for the two phenotypes can result in correlation. Metabolic pathways. The metabolic pathways from gene to phenotype are complex and varied, but the causes of correlation amongst attributes lie within them. Developmental and environmental factors. Multiple phenotypes may be affected by the same factors. There are many phenotypic attributes correlated with age, and so height, weight, caloric intake, endocrine function, and more all have a correlation. A study looking for other common factors must rule these out first. Correlated genotypes and selective pressures. Differences between subgroups in a population, between populations, or selective biases can mean that some combinations of genes are overrepresented compared with what would be expected. While they may not have a significant influence on each other, there may still be a correlation between them, especially when certain genotypes are not allowed to mix. In the process of genetic divergence or having already undergone it can have different characteristic phenotypes, 71, which means that when considered together, a correlation appears. Phenotype qualities in humans that predominantly depend on ancestry also produce correlations of this type. This can also be observed in dog breeds where several physical features make up the distinctness of a given breed, and are therefore correlated. 72. Assortative mating, which is a sexually selective pressure to mate with a similar phenotype, can result in genotypes remaining correlated more than would be expected. Mendelian inheritance. Mendelism is a type of biological inheritance following the principles originally proposed by Gregor Mendel in 1865 and 1866, rediscovered in 1900 by Hugo de Vries and Carl Correns, and later popularized by William Bateson. 1. These principles were initially controversial. Mendel's theories were integrated with the Boveri-Sutton chromosome theory of inheritance by Thomas Hunt Morgan in 1915. They became the core of classical genetics. Ronald Fisher's ideas with the theory of natural selection in his 1930 book The Genetical Theory of Natural Selection, putting evolution onto a mathematical footing and forming the basis for population genetics within the modern evolutionary synthesis. 2. Gregor Mendel, the Moravian Augustinian friar who founded the modern science of genetics. History. The principles of Mendelian inheritance were named for and first derived by Gregor Johann Mendel, 3, a 19th-century Moravian monk who formulated his ideas after conducting simple hybridization experiments with pea plants, Pisum sativum, he had planted in the garden of his monastery. Between 1856 and 1863, Mendel cultivated and tested some 5,000 pea plants. From these experiments, he induced two generalizations which later became known as Mendel's principles of heredity or Mendelian inheritance. Experiments in a two-part paper, Versuche über Pflanzen Hybriden, Experiments on Plant Hybridization, 5 that he presented to the Natural History Society of Brno on the 8th of February and the 8th of March 1865, and which was published in 1866, 3, 6, 7, 8. Mendel's results were at first largely ignored. Although they were completely unknown to biologists of the time, they were not seen as generally applicable, even by Mendel himself, who thought they only applied to certain categories of species or traits. The principles of inheritance were named for and first derived by Gregor Johann Mendel, 3, a 19th-century Moravian monk who formulated his ideas after conducting simple hybridization experiments with pea plants, Pisum sativum, he had planted in the garden of his monastery. 4. Between 1856 and 1863, Mendel cultivated and tested some 5,000 pea plants. From these experiments, he induced two generalizations which later became known as Mendel's principles of heredity or Mendelian inheritance. He described his two-part paper, Versuche über Pflanzen Hybriden, Experiments on Plant Hybridization, 5 that he presented to the Natural History Society of Brno on the 8th of February and the 8th of March 1865, and which was published in 1866, 3, 6, 7, 8. Mendel's results were at first largely ignored. Although they were not completely unknown to biologists of the time, they were not seen as generally applicable, even by Mendel himself, who thought they only applied to certain categories of species or traits. Understanding their significance was the importance attached by 19th-century biologists to the apparent blending of many inherited traits in the overall appearance of the progeny, now known to be due to multi-gene interactions, in contrast to the organ-specific binary characters studied by Mendel. 4. 1900, however, his work was rediscovered by 3 European scientists, Hugo de Vries, Karl Correns, and Erich von Schermach. The exact of the rediscovery has been debated. De Vries published first on the subject, mentioning Mendel in a footnote, while Correns pointed out Mendel's priority after having read de Vries' paper and realizing that he himself did not have priority. De Vries may not have acknowledged truthfully how much of his knowledge of the laws came from his own work and how much came only after reading Mendel's paper. Later scholars have accused von Schermach of not truly understanding the results at all. 9, 10. Regardless, the rediscovery made Mendelism an important but controversial theory. Its most promoter in Europe was William Bateson, who coined the terms genetics and allele to describe many of its tenets. 11. All of heredity was contested by other biologists because it implied that heredity was discontinuous, in opposition to the apparently continuous variation observable for many traits. 12. Many biologists also dismissed the theory because they were not sure it would apply to all species. However, work by biologists and statisticians such as Ronald Fisher showed that if multiple Mendelian factors were involved in the expression of an individual trait, they could produce the diverse results observed, thus demonstrating that Mendelian genetics is compatible with natural selection. 4. Thomas Hunt Morgan and his assistants later integrated Mendel's theoretical model with the chromosome theory of inheritance, in which the chromosomes of cells were thought to hold the actual hereditary material, and created what is now known as classical genetics, a highly successful foundation which eventually cemented Mendel's place in history. 3. 11. Mendel's findings allowed scientists such as Fisher and J.B.S. Haldane to predict the expression of traits on the basis of mathematical probabilities. An important aspect of Mendel's success can be traced to his decision to start his crosses only with plants he demonstrated were true breeding. 4. He only measured discrete binary characteristics, such as color, shape, and position of the seeds, rather than quantitatively variable characteristics. He expressed his results numerically and subjected them to statistical analysis. His method of data analysis and his large sample size gave credibility to his data. He had sight to follow several successive generations P, F1, F2, F3 of pea plants and record their variations. Finally, he performed test crosses, backcrossing descendants of the initial hybridization to the initial true breeding lines to reveal the presence and proportions of recessive characters. He expressed his results numerically and subjected them to statistical analysis. His method of data analysis and his large sample size gave credibility to his data. He had the foresight to follow several successive generations P, F1, F2, F3 of pea plants and record their variations. Finally, he performed test crosses, backcrossing descendants of the initial hybridization to the initial true breeding lines to reveal the presence and proportions of recessive characters. 15. Inheritance tools. Punnett squares. Punnett square, a well-known genetics tool that was created by an English geneticist, Reginald Punnett, which can visually demonstrate all the possible genotypes that an offspring can receive, given the genotypes of their parents. 16, 17, 18. Each parent carries 2 alleles, which can be shown on the top and the side of the chart, and each contribute one of them towards reproduction at a time. Each of the squares in the middle demonstrates the number of times each pairing of parental alleles could combine to make potential offspring. Using probabilities, one can then determine which genotypes the parents can create, and at what frequencies they can be created. 16, 18. example, if two parents both have a heterozygous genotype, then there would be a 50% chance for their offspring to have the same genotype, and a 50% chance they would have a homozygous genotype. Specific attribute to identical alleles, the 50% would be halved to 25% to account for each type of homozygote, whether this was a homozygous dominant genotype or a homozygous recessive genotype. 16, 17, 18. Pedigrees. Pedigrees are visual tree-like representations that demonstrate exactly how alleles are being passed from past generations to future ones. 19. Also provide a diagram displaying each individual that carries a desired allele, and exactly which side of inheritance it was received from, whether it was from their mother's side or their father's side. Are all used to aid researchers in determining the inheritance pattern for the desired allele, because they share information such as the gender of all individuals, the phenotype, a predicted genotype, the potential sources for the alleles, and also based its history, how it could continue to spread in the future generations to come. By using pedigrees, scientists have been able to find ways to control the flow of alleles over time, so that alleles that act problematic can be resolved upon discovery. 20. Mendel's genetic discoveries. 5 parts of Mendel's discoveries were an important divergence from the common theories at the time and were the prerequisite for the establishment of his rules. 1. Character unitary, that is, they are discrete, e.g., purple versus white, tall versus dwarf. There is no medium-sized plant or light purple flower. 2. Genetic characteristics have alternate forms, each inherited from one of two parents. Today these are called alleles. 1. Characters are unitary, that is, they are discrete, e.g., purple versus white, tall versus dwarf. There is no medium-sized plant or light purple flower. 2. Genetic characteristics have alternate forms, each inherited from one of two parents. Today these are called alleles. 3. One allele is dominant over the other. The phenotype reflects the dominant allele. 4. Gametes are created by random segregation. Heterozygotic individuals produce gametes with an equal frequency of the two alleles. 5. Different traits. Today these are called alleles. 1. Characters are Timon were the prerequisite. 1. Characters are unitary, that is. 5 parts of Mendel's discoveries were an important divergence from the common theories at the time and were the prerequisite for the establishment of his rules. 1. Characters are unitary, that is, they are discrete, e.g., purple versus white, tall versus dwarf. There is no medium-sized plant or light purple flower. 2. Genetic characteristics have alternate forms, each inherited from one of two parents. Today these are called alleles. 3. One allele is dominant over the other. The phenotype reflects the dominant allele. 4. Gametes are created by random segregation. Heterozygotic individuals produce gametes with an equal frequency of the two alleles. 5. Different traits have independent assortment. In modern terms, genes are unlinked. According to customary terminology, the principles of inheritance discovered by Gregor Mendel are here referred to as Mendelian laws, although today's geneticists also speak of Mendelian rules or Mendelian principles, 21-22, as there are many exceptions summarized under the collective term non-Mendelian inheritance. The laws were initially formulated by the geneticist Thomas Hunt Morgan in 1916. 23. Characteristics Mendel used in his experiments, 24. P1 generation. The dominant allele for purple red flower hides the phenotypic effect of the recessive allele for white flowers. F2 generation. The recessive trait from the P generation phenotypically reappears in the individuals that are homozygous with the recessive genetic trait. Myosotis. Color and distribution of colors are inherited independently. 25. Mendel selected experiment the following characters of pea plants. Form of the ripe seeds, round or roundish, surface shallow or wrinkled, color of the following characters of pea plants. Form of the ripe seeds, round or roundish, surface shallow or wrinkled, color of the seed coat white, gray, or brown, with or without violet spotting, color of the seeds and cotyledons, yellow or green. Flower color, white or violet red. Form of ripe pods, simply inflated, not contracted, or constricted between the seeds and wrinkled. Color of the unripe pods, yellow or green. Position of the flowers, axial or terminal. Length of the stem, 26. Crossed purebred white flower and purple flower pea plants, the parental or P generation, by artificial pollination, the resulting flower color was not a blend. Rather than being a mix of the two, the offspring in the first generation, F1 generation, were 25. Mendel selected for the experiment the following characters of pea plants. Form of the ripe seeds, round or roundish, surface shallow or wrinkled, color of the seed coat white, gray, or brown, with or without violet spotting, color of the seeds and cotyledons, yellow or green. Flower color, white or violet red. Form of the ripe pods, simply inflated, not contracted, or constricted between the seeds and wrinkled. Color of the unripe pods, yellow or green. Position of the flowers, axial or terminal. Length of the stem, 26. When he crossed purebred white flower and purple flower pea plants, the parental or P generation, by artificial pollination, the resulting flower color was not a blend. Rather than being a mix of the two, the offspring in the first generation, F1 generation, were all purple flowered. Therefore, he called this biological trait dominant. Self-fertilization in the uniform-looking F1 generation, he obtained both colors in the F2 generation with a purple flower to white flower ratio of 3:1. In some of the other characters also one of the traits was dominant. He then conceived the idea of heredity units, which he called hereditary factors. But there are alternative forms of factors, now called genes, that account for variations in inherited characteristics. For example, the gene for flower color in pea plants exists in two forms, one for purple and the other for white. The alternative forms are now called alleles. For each trait, an organism inherits two alleles, one from each parent. These alleles may be the same or different. An organism that has two identical alleles for a gene is said to be homozygous for that gene and is called a homozygote. An organism that has two different alleles for a gene is said to be heterozygous for that gene and is called a heterozygote. Mendel hypothesized that allele pairs separate randomly, or segregate, from each other during the production of the gametes in the seed plant, egg cell, and the pollen plant, sperm. Because allele pairs separate during gamete production, a sperm or egg carries only one allele for each inherited trait. When sperm and egg unite at fertilization, each contributes its allele, restoring the paired condition in the offspring. He also found that each pair of alleles segregates independently of the other pairs of alleles during gamete formation. The type of an individual is made up of the many alleles it possesses. The phenotype is the result of the expression of all characteristics that are genetically determined by its alleles as well as by its environment. The presence of an allele does not mean that the trait will be expressed in the individual that possesses it. If two allele inherited pair differ, the heterozygous condition, then one determines the organism's appearance and is called the dominant allele, the other has no noticeable effect on the organism's appearance and is called the recessive allele. Mendel's laws of inheritance. Law definition. Law of dominance and uniformity. Some alleles are dominant while others are recessive. An organism with at least one dominant allele will display the effect of the dominant allele. 27. Law of segregation. During gamete formation, the alleles for each gene segregate from each other so that each gamete carries only one allele for each gene. Law of independent assortment. Genes of different traits can segregate independently during the formation of gametes. Mendel's laws of inheritance. Law of dominance and uniformity. Generation. All individuals have the same genotype and same phenotype expressing the dominant trait red. F2 generation. The phenotypes in the second generation show a 3:1 ratio. 5% are homozygous with the dominant trait, 50% are heterozygous genetic carriers of the recessive trait, 25% are homozygous with the recessive genetic trait and expressing the recessive character. In Mirabilis jalapa and Antirrhinum majus are examples for intermediate inheritance. 29. As seen in the F1 generation, heterozygous plants have light pink flowers, a mix of red and white. The F2 generation shows a 1:1 ratio of red, light pink, white. If two parents are made of each other who differ in one genetic characteristic for which they are both homozygous, each purebred, all offspring in the first generation, F1, are equal to the examined characteristic in genotype and phenotype showing the dominant trait. This uniformity rule or reciprocity rule applies to all individuals of the F1 generation. 30. Of dominant inheritance discovered by Mendel states that in a heterozygote the dominant allele will cause the recessive allele to be masked, that is, not expressed in the phenotype. Only if an individual is homozygous with respect to the recessive allele will the recessive trait be expressed. Therefore, a cross between a homozygous dominant and a homozygous recessive organism yields a heterozygous organism whose phenotype displays only the dominant trait. The F1 offspring of Mendel's P crosses always looked like one of the two parental varieties. In the situation of complete dominance, the dominant allele had the same phenotypic effect whether present in one or two copies. But for some characteristics, the F1 hybrids have an appearance in between the phenotypes of the two parental varieties. Thus between 2-4 o'clock Mirabilis jalapa plants shows an exception to Mendel's principle, called incomplete dominance. Flowers of heterozygous plants have a phenotype somewhere between the two homozygous genotypes. In case of intermediate inheritance, incomplete dominance, in the F1 generation Mendel's principle of uniformity in genotype and phenotype applies as well. Research about intermediate inheritance was done by other scientists. The first was Carl Correns with his studies about Mirabilis jalapa. 28, 31, 32, 33, 34. Law of segregation of genes. Square for one of Mendel's pea plant experiments, self-fertilization of the F1 generation. Law of segregation of genes applies when two individuals, both heterozygous for a certain trait are crossed, for example, hybrids of the F1 generation. The F2 generation differ in genotype and phenotype so that the characteristics of the grandparents' P generation regularly occur again. In a dominant recessive X, an average of 25% are homozygous with the dominant trait, 50% are heterozygous showing the dominant trait in the phenotype, genetic carriers, 25% are homozygous with the recessive trait and therefore express the recessive trait in the phenotype. The phenotypic ratio is 1:1, and the phenotypic ratio is 3:1. In the example, the capital B represents the dominant allele for purple blossom and lowercase b represents the recessive allele for white blossom. The pistil plant and the pollen plant are both F1 hybrids with genotype BB. Each has one allele for purple and one allele for white. In the offspring, in the F2 plants in the Punnett square, 3 combinations are possible. The genotypic ratio is 1bb, 2b flat, 1bb. The phenotypic ratio of plants with purple blossoms to those with white blossoms is 3:1 due to the dominance of the allele for purple. And with homozygous, BB, or white flowered like one of the grandparents in the P generation. In cases of incomplete dominant, segregation of alleles takes place in the F2 generation, but here also the phenotypes show a ratio of 1:1, as the heterozygous are different in phenotype from the homozygous because the genetic expression of one allele compensates the missing expression of the other allele only partially. This results in an intermediate inheritance which was later described by other scientists. In some literature sources, the principle of segregation is cited as the first law. Nevertheless, Mendel did his crossing experiments with heterozygous plants after obtaining these hybrids by crossing 2 purebred plants, discovering the principle of dominance and uniformity first. 3527. Molecular proof of genes was subsequently found through observation of meiosis by 2 scientists independently, the German botanist Oskar Hertwig in 1876, and the Belgian zoologist Eduard van Benden in 1883. Most alleles are located in chromosomes in the cell nucleus. Paternal and maternal chromosomes get separated in meiosis because during spermatogenesis the chromosomes are segregated on the 4 sperm cells that arise from one mother sperm cell, and during oogenesis the chromosomes are distributed between the polar bodies in the egg cell. Every individual organism contains 2 alleles for each trait. They segregate separate during meiosis such that each gamete contains only one of the alleles. 36. When the gametes unite in the zygote, the alleles, one from the mother, one from the father, get passed on to the offspring. Offspring thus receives a pair of alleles for a trait by inheriting homologous chromosomes from the parent organisms, one allele for each trait from each parent. 6. Heterozygous individuals with the dominant trait in the phenotype are genetic carriers of the recessive trait. Law of independent assortment. Segregation and independent assortment are consistent with the chromosome theory of inheritance. When the parents are homozygous for 2 different genetic traits, LLSS and LLSSP, their children in the F1 generation are heterozygous at both loci and only show the dominant phenotypes, LLSSP. P generation, each parent possesses one dominant and one recessive trait, purebred, homozygous. In this example, solid coat color is indicated by S. Dominant piebald spotting by SP, recessive, while fur length is indicated by L, short, dominant, or L, long, recessive. All individuals are equal in genotype and phenotype. In the combinations of coat color and fur length occur. 9 are short-haired with solid color, 3 are short-haired with spotting, 3 are long-haired with solid color and 1 is long-haired with spotting. The traits are inherited independently, so that new combinations can occur. Average number ratio of phenotypes 9:3:3:1:37. For example, 3 pairs of homologous chromosomes allow 8 possible combinations, all equally likely to move into the gamete during meiosis. This is the main reason for independent assortment. Equation to determine the number of possible combinations given the number of homologous pairs equals 2x, x equals number of homologous pairs. The law of independent assortment proposes alleles for separate traits are passed independently of one another. 3835. That is, the biological selection of an allele for one trait has nothing to do with the selection of an allele for any other trait. Mendel found support for this law in his dihybrid cross experiments. In his monohybrid crosses, an idealized 3:1 ratio between dominant and recessive phenotypes resulted. In dihybrid crosses, however, he found a 9:3:3:1 ratios. This shows that each of the 2 alleles is inherited independently from the other, with a 3:1 phenotypic ratio for each. Independent assortment occurs in eukaryotic organisms during meiotic metaphase and produces a gamete with a mixture of the organism's chromosomes. The physical base of the independent assortment of chromosomes is the random orientation of each bivalent chromosome along the metaphase plate with respect to the other bivalent chromosomes. Along with crossing over, independent assortment increases genetic diversity by producing novel genetic combinations. There are many deviations from the principle of independent assortment due to genetic linkage. 6 chromosomes in a normal diploid human cell, half are maternally derived from the mother's egg and half are paternally derived from the father's sperm. This sexual reproduction involves the fusion of 2 haploid gametes, the egg and sperm, to produce a zygote and a new organism, in which every cell has 2 sets of chromosomes diploid. During gametogenesis, the normal complement of 46 chromosomes needs to be halved to 23 to ensure that the resulting haploid gamete can join with another haploid gamete to produce a diploid organism. Independent assortment, the chromosomes that result are randomly sorted from all possible maternal and paternal chromosomes. Zygotes end up with a mix instead of a predefined set from either parent. Chromosomes are therefore considered assorted independently. As such, the zygote can end up with any combination of paternal or maternal chromosomes. For human History of Science Portal. Biology Portal. Gametes with 23 chromosomes, the number of possibilities is 223 or 8,388,608 possible combinations. 39. This contributes to the genetic variability of progeny. Additionally, the recombination of genes has important implications for many evolutionary processes. 40, 41, 42. A Mendelian one whose inheritance follows Mendel's principles, namely, the trait depends only by history for human. History of Science Portal. Biology Portal. Gametes, with 23. History of Science Portal. Because zygotes end up with a mix instead of a predefined set from either parent, chromosomes are therefore considered assorted independently. As such, the zygote can end up with any combination of paternal or maternal chromosomes. For human. History of Science Portal. Biology Portal. Gametes, with 23 chromosomes, the number of possibilities is 223 or 8,388,608 possible combinations. 39. This contributes to the genetic variability of progeny. Generally, the recombination of genes has important implications for many evolutionary processes. 40, 41, 42. A Mendelian one whose inheritance follows Mendel's principles, namely, the trait depends only on a single locus, whose alleles are either dominant or recessive. Many traits are inherited in a non-Mendelian fashion. 43. Mendel himself warned that care was needed in extrapolating his patterns to other organisms or traits. Indeed, many organisms have traits whose inheritance works differently from the principles he described. These traits are called non-Mendelian. 44, 45. For example, Mendel focused on traits whose genes have only two alleles, such as A and A. However, many genes have more than two alleles. He also focused on traits determined by a single gene. But some traits, such as height, depend on many genes rather than just one. Traits dependent on multiple genes are called polygenic traits. Principle that allele and genotype frequencies in a population will remain constant over generations in the absence of other evolutionary influences. The Hardy-Weinberg law was developed independently by an English mathematician, G. H. Hardy, and a German doctor, Wilhelm Weinberg. 1-2. This concept is also known as the Hardy-Weinberg equilibrium, Hardy-Weinberg theorem or Hardy-Weinberg principle. Sometimes Weinberg's name is placed first. The law is a foundation of population genetics, and it is still taught to students today. 3. It states that the proportions of alleles of all genes in any population will remain the same unless disturbed. That applies to all loci on all chromosomes in the population. Possible perturbations are diamond suit gene mutation, diamond suit natural selection, diamond suit small population size where random effects like genetic drift and inbreeding may occur. HW populations are assumed to be infinite in size. Diamond suit assortative mating instead of random mating. In effect, this would split the population down into small groups, see item above. Diamond suit migration into or out of the population under study. It follows that any systematic change in the frequency of alleles in a population must be due to the effect of one or more of these causes. Of course, like all aspects of Mendelian inheritance, the expected proportions of alleles are probabilities. It was for that reason that statistical tests of significance, such as standard errors, were developed. Although all changes must be due to perturbations, not all perturbations lead to changes. The case is balancing selection, such as heterozygote advantage, heterosis, the heterozygote at a locus is fitter than either homozygote. 4, 5, 6. Balancing selection leads to an equilibrium population with Hardy-Weinberg proportions. Generalization. Law permits the prediction of genotype frequencies from a knowledge of gene frequencies. If alleles are the proportions P and Q, the three zygotic types A, and R in the proportions P2, 2PQ, Q2. Thus, equations can be written in terms of allele frequencies, and hypotheses about how phenotypes are inherited can be tested from population data. Hardy-Weinberg principle. In population genetics, the Hardy-Weinberg principle, also known as the Hardy-Weinberg equilibrium, model, theorem, or law, states that allele and genotype frequencies in a population will remain constant from generation to generation in the absence of other evolutionary influences. These influences include mate choice, assortative mating, natural selection, sexual selection, mutation, gene flow, meiotic drive, genetic hitchhiking, population bottleneck, founder effect, inbreeding and outbreeding depression. Weinberg proportions for two alleles. The horizontal axis shows the two allele frequencies P and Q and the vertical axis shows the expected genotype frequencies. Each line shows one of the three possible genotypes. In the simplest case of two alleles denoted A and A with frequencies FA equals P and FA equals Q, respectively, the expected genotype frequencies under random mating are FA equals P2 for the AA homozygotes, FA equals Q2 for the A homozygotes, and FA equals 2PQ for the heterozygotes. Selection, mutation, genetic drift, or other forces, allele frequencies P and Q are constant between generations, so equilibrium is reached. The principle is named after G. H. Hardy and Wilhelm Weinberg, who first demonstrated it mathematically. Hardy's paper was focused on debunking the view that a dominant allele would automatically tend to increase in frequency, a view possibly based on a misinterpreted question at a lecture 1. Today, tests for Hardy-Weinberg genotype frequencies are used primarily to test for population stratification and other forms of non-random mating. Derivation. Consider a population of monoecious diploids, where each organism produces male and female gametes at equal frequency, and has 2 alleles at each gene locus. We assume that the population is so large that it can be treated as infinite. Organisms reproduce by random union of gametes, the gene pool population model. A locus in population has 2 alleles, A and A, that occur with initial frequencies F0 A equals P and F0 A equals Q, respectively. Note 1 allele frequencies at each generation are obtained by pooling together the alleles from each genotype of the same generation according to the expected contribution from the homozygote and heterozygote genotypes, which are 1 and 1/2, respectively. 1, 2. Length of P, Q corresponds to allele frequencies, here P equals 0.6, Q equals 0.4. Then area of rectangle represents genotype. Frequencies thus. A equals 0.36, 0.48, 0.16. The diploids to form genotypes for the next generation can be shown in a Punnett square, where the proportion of each genotype is equal to the product of the row and column allele frequencies from the current generation. Table 1, Punnett square for Hardy-Weinberg. Females A, P, A, Q. Males A, P, A, P2, A, PQ. A, Q, A, QP, A, Q2. Sum of the entries is P2 plus 2 PQ plus Q2 equals 1, as the genotype frequencies must sum to 1. Note again that as P plus Q equals 1, the binomial expansion of P plus Q, 2 equals P2 plus 2 PQ plus Q2 equals 1 gives the same relationships. Element of the Punnett square or the binomial expansion, we obtain the expected genotype proportions among the offspring after a single generation. 3, 4, 5. These frequencies define the Hardy-Weinberg equilibrium. It should be mentioned that type frequencies after the first generation need not equal the genotype frequencies from the initial generation, e.g. F1 A does not equal F0 AA. How the genotype frequencies for all future times will equal the Hardy-Weinberg frequencies, e.g. featuring A equals F1 A for t greater than 1. This follows genotype frequencies generation depend only on the allele frequencies of the current generation which, as calculated by equations 1 and 2, are preserved from the initial generation. For the more general case of dioecious diploids, organisms are either male or female that reproduce by random mating of individuals. It is necessary to calculate the genotype frequencies from the 9 possible matings between each parental genotype AA, A, and A in either sex, weighted by the expected genotype contributions of each such mating. 2. Equivalently, one considers the 6 unique diploid-diploid combinations and constructs a Punnett square for each, so as to calculate its contribution to the next generation's genotypes. Contributions are weighted according to the probability of each diploid-diploid combination, which follows a multinomial distribution with k equals 3. For example, the probability of the mating combination AA, A is 2 feet AA featuring A, and it can only result in the A genotype, 0, 1, 0. Overall, the resulting genotype frequencies are calculated as before. One can show that the allele frequencies at time t plus 1 equal those at time t, and so, are constant in time. Similarly, the genotype frequencies depend only on the allele frequencies, and so, after time t equals 1 are also constant in time. Monoecious or dioecious organisms, either the allele or genotype proportions are initially unequal in either sex. It can be shown that constant proportions are obtained after one generation of random mating. If dioecious organisms are like a gene locus is located on the X chromosome, it can be shown that if the allele frequencies are initially unequal in the two sexes, e.g., XX females and XY males, as in humans, F, A in the heterogametic sex chases F, A in the homogametic sex of the previous generation, until an equilibrium is reached at the weighted average of the two initial frequencies. Deviations from Hardy-Weinberg equilibrium. The 7 assumptions underlying Hardy-Weinberg equilibrium are as follows. 3. Organisms are diploid. Only sexual reproduction occurs. Generations are non-overlapping. Mating is random. Population. Only sexual reproduction occurs. The 7 assumptions underlying Hardy-Weinberg equilibrium are as follows. 3. Organisms are diploid. Only sexual reproduction occurs. Generations are non-overlapping. Mating is random. Population size is infinitely large. Allele frequencies are equal in the sexes. There is no migration, gene flow, admixture, mutation or selection. Violations of the Hardy-Weinberg assumptions can cause deviations from expectation. How this affects the population depends on the assumptions that are violated. Random mating states the population will have the given genotypic frequencies, called Hardy-Weinberg proportions, after a single generation of random mating within the population. When the random mating assumption is violated, the population will not have Hardy-Weinberg proportions. A common cause of non-random mating is inbreeding, which causes an increase in homozygosity for all genes. If the population violates one of the following 4 assumptions, the population may continue to have Hardy-Weinberg proportions each generation, but the allele frequencies will change over time. Selection, in general, causes allele frequencies to change, often quite rapidly. Wild directional selection eventually leads to the loss of all alleles except the favored one, unless one allele is dominant, in which case recessive alleles can survive at low frequencies. Some forms of selection, such as balancing selection, lead to equilibrium without loss of alleles. The HWP states the population. Random mating. The HWP states the population will have the given genotypic frequencies, called Hardy-Weinberg proportions, after a single generation of random mating within the population. When random mating assumption is violated, the population will not have Hardy-Weinberg proportions. A common cause of non-random mating is inbreeding, which causes an increase in homozygosity for all genes. Population violates one of the following four assumptions. The population may continue to have Hardy-Weinberg proportions each generation, but the allele frequencies will change over time. Selection, in general, causes allele frequencies to change, often quite rapidly. Wild directional selection eventually leads to the loss of all alleles except the favored one, unless one allele is dominant, in which case recessive alleles can survive at low frequencies. Some forms of selection, such as balancing selection, lead to equilibrium without loss of alleles. Mutation will have a very subtle effect on allele frequencies through the introduction of new allele into a population. Mutation rates are of the order 10⁻⁴ to 10⁻8, and the change in allele frequency will be, at most, the same order. Recurrent mutation will maintain alleles in the population, even if there is strong selection against them. Migration genetically links two or more populations together. In general, allele frequencies will become more homogeneous among the populations. Some models for migration inherently. Mutation will have a very. Selection, in general, causes allele frequencies to change, often quite rapidly. Wild directional eventually leads to the loss of all alleles except the favored one, unless one allele is dominant, in which case recessive alleles can survive at low frequencies. Some forms of selection, such as balancing selection, lead to equilibrium without loss of alleles. Mutation will have a very subtle effect on allele frequencies through the introduction of new allele into a population. Mutation rates are of the order 10⁻⁴ to 10⁻8, and the change in allele frequency will be, at most, the same order. Recurrent mutation will maintain alleles in the population, even if there is strong selection against them. Migration genetically links two or more populations together. In general, allele frequencies will become more homogeneous among the populations. Some modelically links two or more populations together. In general, allele frequencies will become more homogeneous among the populations. Migration genetically links two or more populations together. In general, allele frequencies will become more homogeneous among the populations. Some models for migration inherently include non-random mating, Walland effect, for example. For those models, the Hardy-Weinberg proportions will normally not be valid. Small population size can cause a random change in allele frequencies. This is due to a sampling effect, and is called genetic drift. Sampling effects are most important when the allele is present in a small number of copies. In real-world genotype data, deviations from Hardy-Weinberg equilibrium may be a sign of genotyping error. 4, 5, 6. Sex linkage. Where the gene linked, the heterogametic sex, e.g., mammalian males, avian females, have only one copy of the gene and are termed homozygous, while the homogametic sex, e.g., human females, have two copies. Genotype frequencies at equilibrium are P and Q for the heterogametic sex but P2, 2PQ and Q2 for the homogametic sex. For example, in humans red-green colorblindness is an X-linked recessive trait. In Western Europeans, the trait affects about 1 in 12, Q equals 0.083, whereas it affects about 1 in 200 females, 0.005, compared to Q2 equals 0.007, very close to Hardy-Weinberg proportions. If the population of males and females with a different allele frequency in each subpopulation, males or females, the allele frequency of the male population in the next generation will follow that of the female population because each son receives its X chromosome from its mother. The population converges on equilibrium very quickly. Generalizations. The simple derivation above can be generalized for more than two alleles in polyploidy. Generalization for more than two alleles. Punnett square for 3 allele case left and 4 allele case right. White areas are homozygotes. Colored areas are heterozygotes. Consider an extra allele frequency, R. This is the binomial expansion of P plus Q2, and thus the 3 allele case is the trinomial expansion of P plus Q plus R2. More generally, consider the alleles A1, and given by the allele frequencies P1 to Pn. Giving for all homozygotes. And for all heterozygotes. Generalization for polyploidy. The Hardy-Weinberg principle may also be generalized to polyploid systems, that is, for organisms that have more than 2 copies of each chromosome. Consider again only 2 alleles. The diploid case is the binomial expansion of. And therefore the polyploid case is the binomial expansion of. Where C is the ploidy, for example with tetraploid, C equals 4. Table 2, expected genotype. Frequencies for tetraploidy. Genotype frequency. Quadruple A quadruple A quadruple A quadruple A quadruple A. Whether the organism is a true tetraploid or an amphidiploid will determine how long it will take for the population to reach Hardy-Weinberg equilibrium. Complete generalization. For distinct alleles in ploids, the genotype frequencies in the Hardy-Weinberg equilibrium are given by individual terms in the multinomial expansion of. Significance tests for deviation. deviation from the HWP is generally performed using Pearson's chi-squared test, using the observed genotype frequencies obtained from the data and the expected genotype frequencies obtained using the HWP. For cysts where there are numbers of alleles, this may result in data with many empty possible genotypes and low genotype counts, because there are often not enough individuals present in the sample to adequately represent all genotype classes. If it's the case, then the asymptotic assumption of the chi-squared distribution will no longer hold, and it may be necessary to use a form of Fisher's exact test, which requires a computer to solve. More number of MCMC methods of testing for deviations from HWP have been proposed. Guo and Thompson, 1992, Wigginton et al. 2005. Example chi-squared test for deviation. From E.B. Ford, 1971, on the scarlet tiger moth, for which the phenotypes of a sample of the population were recorded. Genotype-phenotype distinction is assumed to be negligibly small. The null hypothesis is that the population is in Hardy-Weinberg proportions, and the alternative hypothesis is that the population is not in Hardy-Weinberg proportions. Table 3. Example Hardy-Weinberg principal calculation. Phenotype white spotted intermediate little spotting total. Number 146913851612. From this, allele frequencies can be calculated. And so the Hardy-Weinberg expectation is. Pearson's chi-squared test states. There is 1 degree of freedom. Degrees of freedom for test for Hardy-Weinberg proportions are hash genotypes minus hash alleles. The 5% sigma level 1 degree of freedom is 3.84, and since the chi2 value is less than this, the null hypothesis that the population is in Hardy-Weinberg frequencies is not rejected. Fisher's exact test probability tests. Fisher's exact test can be applied to testing for Hardy-Weinberg proportions. Since the test is conditional on the allele frequencies, P and Q, the problem can be viewed as testing for the proper number of heterozygotes. In this way, the hypothesis of Hardy-Weinberg proportions is rejected if the number of heterozygotes is too large or too small. The conditional probabilities of a zygote, given the allele frequencies are given in MI 1980, as where N11, N12, N22 are the observed numbers of the 3 genotypes, and respectively, and N1 is the number of alleles, where. And using one of the examples from MI 1987, we can consider the case where N equals 100, and P equals 0.34. Possible observed heterozygotes and their exact significance level is given in Table 4. Table 4. Example of Fisher's exact test for N equals 100, P equals 0.34. The equivalence tests are developed in order to establish a table like this. Using this table, one must look up a significance level of the test based on the observed number of heterozygotes. For example, if one observed 20 heterozygotes, the significance level for the test is 0.007. As is typical for Fisher's exact test for small samples, the gradation of significance levels is quite coarse. However, a table like this has to be created for every experiment, since the tables are dependent on both N and P. Equivalence tests. These tests are developed in order to establish sufficiently good agreement of the observed genotype frequencies in Hardy-Weinberg equilibrium. Let denote the family of the genotype distributions under the assumption of Hardy-Weinberg equilibrium. The distance between a genotype distribution and Hardy-Weinberg equilibrium is defined by, where is some distance. The equivalence test problem is given by and. Where is a tolerance parameter? If the hypothesis can be rejected, then the population is close to Hardy-Weinberg equilibrium with a high probability. The equivalence tests for the Bialek case are developed among others in Welek 2004. 8. The equivalence tests for the case of multiple alleles are proposed in Ostrovsky 2020. 9. Inbreeding coefficient. The breeding coefficient, see also F statistics, is 1 minus the observed frequency of heterozygotes over that expected from Hardy-Weinberg equilibrium, where the expected value from Hardy-Weinberg equilibrium is given by. For example, for Ford's data above. For 2 alleles, the chi-squared goodness of fit test for Hardy-Weinberg proportions is equivalent to the test for inbreeding. The inbreeding coefficient is unstable as the expected value approaches 0, and thus not useful for rare and very common alleles. 4. Is undefined. History. Mendelian genetics were rediscovered in 1900. However, it remained somewhat controversial for several years as it was not then known how it could cause continuous characteristics. Yule 1902 argued against Mendelism because he thought that dominant alleles would increase in the population. And William E. Castle 1903 showed that without selection, the genotype frequencies would remain stable. 11. Carl Pearson 1903 found one equilibrium position with values of P equals Q equals 0.5. 12. Punnett, unable to counter Yule's point, introduced the problem to G. H. Hardy, a British mathematician, with whom he played cricket. Hardy was a pure mathematician and held applied mathematics in some contempt. His view of biologists' use of mathematics comes across in his 1908 paper where he describes this as very simple. 13. To the I am reluctant to intrude in a discussion concerning matters of which I have no expert knowledge, and I should have expected the very simple point which I wish to make to have been familiar to biologists. However, some remarks of Mr. Udny Yule, to which Mr. R.C. Punnett has called my attention, suggest that it may still be worth making. Suppose that a pair of Mendelian characters, A being dominant, and that in any given generation the number of pure dominants AA, heterozygotes A, and pure recessives A, are ASP, 2Q, R. Suppose that the numbers are fairly large, so that mating may be regarded as random, that the sexes are evenly distributed among the three varieties, and that all are equally fertile. A little mathematical multiplication table type is enough to show that in the next generation the numbers will be as P plus Q, 2, 2P plus Q, Q plus R, sad face Q plus R, 2, or as P1, 2Q1, R1, say. The interesting question is, in what circumstances will this distribution be the same as that in the generation before? It is easy to see that the condition for this is Q2 equals PR. And since Q1, 2 equals P1 R1. Whatever the values of P, Q, and R may be, the distribution will in any case continue unchanged after the second generation. The proof was thus known as Hardy's law in the English-speaking world until 1943, when Kurt Stern pointed out that it had first been formulated independently in 1908 by the German physician Wilhelm Weinberg. 14. William Castle in 1903 also derived the ratios for the special case of equal allele frequencies, and it is sometimes but rarely called the Hardy-Weinberg-Castle law. Derivation of Hardy's equations. Hardy's statement begins with a recurrence relation for the frequencies P, 2Q, and R. These recurrence relations follow from fundamental concepts in probability, specifically independence, and conditional probability. For example, consider the probability of an offspring from the generation being homozygous dominant. Alleles are inherited independently from each parent. A dominant allele can be inherited from a homozygous dominant parent with probability 1, or from a heterozygous parent with probability 0.5. To represent this reasoning in an equation, let represent inheritance of a dominant allele from a parent. Furthermore, let n represent potential parental genotypes in the preceding generation. The same reasoning, applied to the other genotypes, yields the two remaining recurrence relations. Equilibrium occurs when each proportion is constant between subsequent generations. More formally, a population is at equilibrium at generation when solving these equations necessary and sufficient conditions for equilibrium to occur can be determined. Again, consider the frequency of homozygous dominant animals. Equilibrium implies first consider the case, where, and note that it implies that n. Now consider the remaining case, where the final equality holds because the allele proportions must sum to 1. In both cases. It can be shown that the other two equilibrium conditions imply the same equation. Together, the solutions of the three equilibrium equations imply sufficiency of Hardy's condition for equilibrium. Since the condition always holds for the second generation, all succeeding generations have the same proportions. Numerical example. Estimation of genotype distribution. An example computation of the genotype distribution given by Hardy's original equations is instructive. The phenotype distribution from Table 3 above will be used to compute Hardy's initial genotype distribution. Note that the P and Q values used by Hardy are not the same as those used above. As checks on the distribution, compute. And for the next generation, Hardy's equations give. Again as checks on the distribution, compute. And which are the expected values? The reader may demonstrate that subsequent use of the second generation values for a third generation will yield identical results. Estimation of carrier frequency. The Hardy-Weinberg principle can also be used to estimate the frequency of carriers of an autosomal recessive condition in a population based on the frequency of suffers. Let us assume an estimated babies are born with cystic fibrosis. This is about the frequency of homozygous individuals observed in Northern European populations. We can use the Hardy-Weinberg equations to estimate the carrier frequency, the frequency of heterozygous individuals. As is small, we can take P to be 1. We therefore estimate the carrier rate to be, which is about the frequency observed in Northern European populations. This simplified to the carrier frequency being about twice the square root of the birth frequency. Graphical representation. A Definetti diagram representing a distribution of genotype frequencies. It is possible to represent the distribution of genotype frequencies for a biallelic locus within a population graphically using a Definetti diagram. It uses a triangular plot, also known as trilinear, triaxial or ternary plot, to represent the distribution of the three genotype frequencies in relation to each other. It differs from many other such plots in that the direction of one of the axes has been reversed. 16. The curved line in the diagram is the Hardy-Weinberg parabola and represents the state where alleles are in Hardy-Weinberg equilibrium. It is possible to represent the effects of genetic drift, also known as random genetic drift, allelic drift or the right effect. 1 is the change in the frequency of an existing gene variant allele in a population due to random chance. 2. Genetic drift may cause gene variants to disappear completely and thereby reduce genetic variation. 3. It can also cause initially rare alleles to become much more frequent and even fixed. When few copies of an allele exist, the effect of genetic drift is more notable, and when many copies exist, the effect is less notable due to the law of large numbers. In the middle of the 20th century, vigorous debates occurred over the relative importance of natural selection versus neutral processes, including genetic drift. Ronald Fisher, who explained natural selection using Mendelian genetics, held the view that genetic drift plays at most a minor role in evolution, and this remained the dominant view for several decades.